All right, so welcome to the presentation for Chapter 15 of our Anatomy and Physiology textbook, which is concerned with pathways. So let's talk about what a pathway is first so that you know what I mean when I say that word. Um, Basically, it's the series of nerves, nuclei, tracts that a impulse, an action potential, must pass through and be carried along to get to where it's ending up. So this includes both descending and ascending pathways, so both going from the CNS and to the CNS. So we can divide these into sensory pathways and somatic motor pathways. So chapter 16 is the autonomic nervous system chapter that's next. So visceral motor pathways we'll talk about in that chapter, not in this one. So sensory and somatic motor, so general senses and some special ones, and then somatic motor is going to be, of course, skeletal muscle and voluntary movement. So let's talk a little bit about sensory receptors. So receptors are essentially structures with cells that excite and generate what's called a receptor potential under specific sensory conditions. So things like touch receptors are mechanoreceptors. So those have mechanically gated ion channels, which is to say ion channels that are going to open when the membrane of the cell is physically disturbed or poked or prodded. And then we have other kinds of sensory receptors as well. Generally, what a receptor does is take the stimulus, whatever it is, and turn it into a receptor potential. And you notice I'm not saying action potential, and that is because in many cases, although not all, it is not the sensory cell that does the action potential. It's the cells or cell that it excites instead. So to sort of explain what a receptor potential is, I'm going to just go into whiteboard mode really quick and draw something. And so I'll just use my favorite of these colors. So let's take, for example, I don't know, it doesn't really matter. The majority of sensory cells and receptors release glutamate when they are excited by the sensory stimulus that they're designed to detect. And that's really well characterized for some receptor cells and not very well characterized for others. I'm just going to use the example of a cone cell in the eye, but we can use many other ones. So when a light strikes a cone cell, just drawing different parts of it, and then it has this little area right here and its nucleus. Has a little stack of membranous structures that catch the light. So zooming in on the synaptic terminus, when a photon strikes the light-sensitive portion, the outer segment of the cone cell, that generates a receptor potential, which is going to travel down the membrane like so, and cause the release of vesicles of neurotransmitter, and specifically the neurotransmitter glutamate, or glutamic acid. And that's going to cause the fusion of the vesicles with the cell membrane, and that lets the glutamate out into the synapse. And then that's going to bind to receptors on the dendrite of a bipolar cell. So the glutamate binds, and that generates a graded potential, which ultimately excites the bipolar cell. And the bipolar cell has an action potential as a result of this, in pink here, receptor potential. And then let's give our little bipolar cell an action potential, which I'm going to illustrate as being green. So as a result of this glutamate, we get an action potential from that graded potential, which causes the bipolar cell to release its neurotransmitter onto the next cell in the chain, which is the ganglion cell. So this one is an action potential. So that's what I mean when I say receptor potential. I'm talking about the membrane voltage change that allows a receptor cell to turn a non-neural signal into a neural signal. So that's the magic of receptors. That action is called transduction. Turn external stimulus into coded action potentials. Because the brain only knows about action potentials, and that's the only kind of information it can make sense of. So if I want to detect a stimulus, like taste, for example, I want to taste salt or taste sourness. I'm going to detect chemicals in the food I eat using receptors for those chemicals. And then the receptor is going to let go of the chemical, and I'm going to swallow it, and it's gone. So the excitation of the receptor, it's not going to create another flavor molecule and send the flavor molecule to my brain. It's going to translate that flavor sensation into action potentials that my brain can then make sense of. So that's what transduction is. It's kind of magic a little bit. Oopsie, I just want to get out of whiteboard mode. There we go. So that was just a little piece of the puzzle that's sort of missing here. So I wanted to make sure it was explicitly discussed. So we do have, transduction mentioned here, but not much about how that happens. So that's what I was trying to fill in. So sensory impulses are going to travel inwards via a first order sensory neuron up the spinal cord, and they are filtered at the thalamus, meaning the thalamus sorts out which kind of sense it's receiving and where it needs to go in the cortex. And sometimes they just project directly to the cerebral cortex without passing through the thalamus. It depends on what kind of sensation it is. So a sensation is when the arriving impulse strikes the cortex and we become consciously aware of it. So sensation and perception are not the same thing. So sensation turns into perception when we, via our frontal and prefrontal cortex, are made aware of the stimulus. Sensation is the arriving portion. So we have somatic sensory pathways and visceral sensory pathways. I know the example I used previously was a cone cell of the eye. That's a special sense, and we'll get to those in chapter 17 in more detail. But for now, we're talking about some specific pathways that are going to connect our body to our brain. So the origin is, of course, receptors, and these are going to generate receptor potentials. Many of them are glutamatergic, which is glutamate is the neurotransmitter, but not all. So the receptors are all over the place. These can be things like Pacinian corpuscles, which you might remember from chapter 5. They can be things like pain receptors, which are called nociceptors, by the way, or pressure receptors or sensations from Merkel cells, all kinds of stuff in the somatic sensory area. And this includes proprioceptors as well. The destinations via the somatic sensory pathways. For that portion of sensory is the primary sensory cortex, so that is on the post-central gyrus. Remember I talked about a map of your body on that gyrus? You can see that map here. And also the cerebellum, and this is of course for proprioception. The visceral sensory pathways, unsurprisingly, arrive at nuclei in the brainstem, so pons and medulla, and at the diencephalon, so hypothalamus and thalamus. Okay, so let's talk about the idea of a labeled line. It's kind of like a telephone line where a series of telephone wires stretches from one location to my house, and that line, even at its origin, is labeled phone line to Christina's house. Additionally, if a phone call originates from somebody, that line from them to me is labeled phone call from so-and-so because it traveled along a specific pathway, and that pathway has a name and a specific origin and destination. So when I say labeled line in sensory impulses, what I mean is the way that the body knows that you are, for example, sensing pain in your foot, like in this old illustration, is because a pain fiber and a pain receptor are activated here in this part of the foot, and that line is labeled pain foot, specifically that part of the foot. So it's a fiber for pain, and it is specific to this region of the foot. And so this whole entire wire or axon and it's synapsing, and so the second order sensory neuron going up to the brain is labeled pain foot. And so the brain and the thalamus is going to pass that information to the foot area, and then that's made sense of by the rest of your brain to say, oh, okay, the pain is occurring in the foot. So each sensory modality, so pain, cold, etc., is carried by one labeled line. So you might have, there's a lot of sensors in your foot. There's also mechanoreceptors and proprioceptors. So all of those senses coming from those receptors have different labeled lines. They're associated with the same foot area or body area, but they're carrying different information. So an arriving impulse from a mechanoreceptor is not also carrying an arriving impulse from a pain receptor. Those are separate lines. And that's how the brain makes sense of and sorts them. So a link between a receptor and the cortical neuron in the cerebrum. So you can use this idea to understand phantom limb pain, which is one of the things I asked you to listen to a podcast about in the case study. But essentially, since we know that the labeled line is labeled, if you say, lose your leg, gone. The first order sensory neuron, the one that's coming in to the spinal cord, is gone. But the rest of these sensory neurons, so the one originating from the spinal cord and going to the thalamus, so the second order, and the one going from the thalamus to the appropriate cortical location, the third order, those are still intact. So because this part of the line is still intact, it's just the first order sensory neuron that's gone. If this sensory line is stimulated by accident for some reason, it's still going to be directed to the appropriate cortical location. Let's do third order to foot. So if it's mistakenly activated, you're going to have sensations in a leg that's no longer there. And that's explained very thoroughly in the Radiolab podcast that I recommended and had you listen to, so you can go get more details there. But essentially, if part of the labeled line is still intact, that creates the possibility to sense a limb artificially that's not really there at all. So, interpretation of sensory information. This involves the primary sensory areas and then their corresponding association areas, right? So axons are going to carry the sensory information in bundles, so groups, that are traveling to a common or specific destination. So for example, in the hand, we would expect to see the axons of mechanoreceptors, pain receptors, and hot and cold receptors, respectively. All those sensors are carried on different axons, but all of those axons are going to travel together to the hand location because they're about the same region, but they're carrying different modalities. So when the stimulus arrives on the cortex, obviously, it's going to determine the perceived location. So when it arises to my conscious perception, it's because the impulse has stimulated that appropriate part of my somatosensory cortex that is labeled the hand zone, and then I can say, I'm feeling a sensation in my hand. The frequency of the action potentials, so how many per unit time, is going to determine the intensity of the stimulus. So if I have a really intense stimulus, like something's really cold, the action potentials would be really close together. So this is a way we represent action potentials. It's basically crosshatches on a line to show you time, and then each of these represents a spike. So if I had a really cold sensation, it would be looking like that, and if it was just kind of cold, it would look more like that. So this is what we call encoding, meaning it's not good enough to just have an action potential, you have to have a pattern of them that provides information about the amount of the stimulus and other information as well. So how are somatic sensory pathways organized? Well, I talked about the order neurons in the last page, and here we are. So the first order neuron is from receptor to spinal cord. The second order neuron is from the spinal cord to thalamus. And the third order neuron, in most cases, but not always, is from, I can't write R's, the thalamus to cortex. So it's a game of neural telephone where the first order neuron tells the second order neuron, hey, sensation is happening. Also, it's touch. And the second order neuron carries that information to the thalamus, and then the thalamus integrates that and says, okay, so we're receiving this information from this labeled line that is touch specifically, and it's coming from hand. So I'm going to provide a third order neuron that is specific to touch and specific to that hand region, and I'm going to project it up to the hand area of the post-central gyrus. There are some cases where the third order neuron isn't always present, but that's, on a case-by-case basis, so we're not going to talk much about that. Axon collaterals may exist. Remember, every cell only has one axon. So dendrites down here, this is the axon, but the axon could split and go to a couple different places. It's still considered to be one because if there's an action potential originated here, it will guaranteed propagate down both collaterals. There's no way to do one or the other. So that's what collaterals mean, and that's still meeting the one axon rule. So these are going to maybe also send impulses to cerebellum. So if it's proprio receptive information, it would probably also get sent to the cerebellum. The reticular formation might also get activated as well via collaterals. It really just depends on what the sensory modality is and what it means. So the primary somatosensory cortex is expressed in, what's called a sensory homunculus, which means little human. So you'll notice that on this map here, the hand and the face are really big. So is the tongue and the jaw. The rest of the body, torso, back of the head, so not front of face, legs and butt are pretty small comparatively. So this has a smaller of cortical neurons, and this has lots of cortical neurons. So the number of sensory receptors in the area, so you have a lot in your hands, a lot in your face, because our face and our hands are how we interact with the world. And then, you don't really tend to determine information or absorb info from your back very much, so that's smaller by comparison. So the number of sensory receptors is what determines the largeness of the cortical area associated with that body region. So if you put that all together, what you get is a sensory homunculus, which looks like this, which is disturbing. It's a little man with skinny arms and legs and a tiny torso, and then giant hands and a giant face. Interesting. All right, so the major somatic sensory pathways, there are 3, and this is actually why we made you learn the posterior column for your practical, because it is containing stuff that you need to know. So we've got the posterior column pathway, which includes the fasciculus gracilis and the fasciculus cuneatus. Those are referring to which nuclei in various locations those sensory informations will pass through. And then we've got the spinocerebellar tracts, which are going to come from the spinal cord and go to the cerebellum, as their name suggests, and then the spinothalamic tracts, which obviously go from the spinal cord to the thalamus. So with the exception, oh no, sorry, I moved my tablet and it thinks it needs to be vertical. With the exception of the posterior column pathway, all of these are named for their origination point and their destination. And I'm using origination point with an asterisk here because, of course, the spinal cord has to get the information from somewhere, right? So that's going to come from receptors. So we've got the anterior and lateral spinothalamic tracts, and then the spinocerebellar tracts, which are in yellow here. There's an anterior and a posterior, and then the posterior column pathway as a whole. So the spinothalamic pathway contains 2 tracts. I don't know why it says FYI here. You do need to know this. It's just an accidental carryover from copy pasting. So there are 2 tracts, and essentially one of them is going to detect crude touch and pressure from one side of the body or the other. There's some on each side. So this one happens to be the right side of the body. That's what this image happens to be illustrating, but there's ones on the left too, so don't be confused about that. So the anterior spinothalamic tract detects crude touch and pressure sensations. So the feeling of being touched, general touch, so not fine touch like in your hands, but just like, are you wearing clothes and you can feel them? Are you feeling the pressure on your bottom of sitting in a chair? Those kinds of things. And then the lateral spinothalamic tract is for pain and temperature. So 2 different tracts for 2 different modalities. So these are going to be divided accordingly, but they end up at the same sensory or cortical location. So you can also see they both decussate in the spinal cord, specifically in the anterior gray commissure, and then they ascend in their respective tracts, but in parallel. and then they arrive at the same destination. So different modalities, same destination. Anterior, crude touch and deep pressure, lateral, pain and temperature. So one of the consequences of the spinothalamic pathways is that because of the density of neurons, let's go back one, because of the density of sensory neurons in the dorsal gray horn, there's just oodles of them, it's possible for them to accidentally excite each other every now and then. And so if in the process of going somewhere, the spinothalamic pathway accidentally excites a visceral receptor, that can cause what's called referred pain, which is where you're feeling pain that is not in the location of the pain signal. So, for example, as many people know, if you have a heart attack, you can get pain in your left shoulder that radiates down your left arm, and that is a sign that you need to go to the hospital. If you have a kidney stone traveling through your ureter, you might feel side and inguinal pain, and so on and so forth. Gallbladder attacks, which is where your gallbladder is trying to secrete bile, but there's a gallstone in the way, are often detected in the skin here and can even radiate up to the shoulder. And this is just due to cross excitation. And then I already talked about phantom limb pain, which is just excitation in the spinal cord or even above that accidentally excites the pain receptors that are traveling to the cortical area of the phantom limb, even though there is no longer a limb there. So we talked about this already, but it's always good to reinforce. So inappropriate sensations occur when part of a labeled line is stimulated. So maybe in the spinal cord due to the arriving sensory information of some other sense, some neurotransmitter accidentally ends up on this receptor or this the dendrites of this neuron. And so it's like, okay, well, what I do when I get excitation is I just send it upstairs. And so the thalamus is like, there's pain and it's on the leg, let's say. And so you have an ouchy or itchy leg, even though you don't have a leg anymore. So that's what phantom limb pain is. And there are some interesting strategies that one can use to get rid of it or deal with it, which are described in that podcast I mentioned. So referred pain, again, is inaccurate localization, and here we have a more close view of the dorsal horn of the spinal cord, which I mentioned just a moment ago. So if we zoom in and look, we can see that the somatic sensory receiving area and the visceral sensory receiving area are right next to each other, and presumably their surrounding extracellular fluid is shared, so it's possible for accidental excitation. So let's say we have, a visceral sensory event of the nature I described, so heart attack, kidney stone, etc., that is causing visceral pain, and it, accidentally excites a somatic sensory neuron as well, and that causes the sensation of pain in the limb or in the skin when it's really being sensed viscerally, because that's where the tissue damage and the problem is actually occurring. So they stimulate interneurons and end up in the spinothalamic pathway, which you may remember is for pain and temperature, and then they get sent to the appropriate cortical location, which is unfortunately incorrect. So that's what referred pain is. So again, not FYI. The posterior column pathway is for everything else. So fine touch, like for example, hand. So you can determine, for example, via your Merkel cells and very delicate receptors. You can, if someone hands you a piece of genuine silk and a piece of microfiber, you can tell the difference by touching it. You can discriminate tiny bumps on things, the textures of objects, etc. That's what fine touch means. And then pressure, vibration, and of course, proprioception to some extent. So there's proprioception duties that are shared by the posterior column pathway, but also the spinocerebellar pathway. So there's a little bit of overlap in responsibilities there. So what you'll see is that these neurons, the first order neurons, they skip synapsing in the spinal cord, and instead they ascend within the pyramids of the medulla oblongata up to the nucleus gracilis and cuneatus. That's what it means when it says that portion. So remember, I pointed out that the posterior column pathway had these two specific names, and that was nuclei. So that's where these are synapsing. That's why it's called that. And then after either the nucleus gracilis or the nucleus cuneatus, then it decussates within the medulla. And the second order neuron travels up through the midbrain, again to the thalamus. So decussates in medulla oblongata, not in spinal cord. So fine touch, pressure, vibration, and proprioception, and they do decussate, so one side of the body is sensed and perceived by the opposite brain area. Okay, spinocerebellar pathways. These are a little bit easier because there's not a first, second, and third order neuron. There's only two, first order and second order. And spinocerebellar makes it nice and easy to figure out where the destination is. So from the spinal cord to the cerebellum. So we have first order and second order. And these are subconscious proprioception, so limb position and posture stuff. So these contain information received from joints, muscles via the muscle spindle, tendons via the Golgi tendon organ, and joints via joint capsule receptors. So this is going to tell your cerebellum, is my elbow flexed? Is it flexed the appropriate amount for the movement I'm trying to do? Or, is my muscle being stretched, for example? Those are all going to be sent to the cerebellum for proprioception, but not in a way that you experience consciously. So you're not going to be like, oh, I'm experiencing the sensation that my elbow is at a 45-degree angle. No, it's just going to go to the cerebellum to provide information to the cerebellum about movement while you're doing it. So the anterior and posterior. One decussates in the anterior great commissure of the spinal cord. The other one does not, and it stays on the same side, and that is going to be here. So you can see the posterior spinocerebellar tract does not decussate, so that makes it, let's erase these FYIs. So that's going to make the posterior spinocerebellar tract what's called ipsilateral, and the anterior one contralateral. So ipsilateral means same side, so it stays on the same side, and contralateral means it decussates. So we can see Golgi tendon organs, muscle spindles, and joint capsule receptors. So we have trunk and lower limb and trunk and lower limb. So posture for trunk and walking and standing for lower limb. And we have both same side and opposite side components to it. So this is an FYI, so this one's okay. You don't need to know the cuneo-cerebellar or rostral-cerebellar, spinocerebellar pathways. These are not listed in your textbook, that's why. So these are from the trunk and upper limbs. And the upper limbs also get their own pathway for another thing, which we'll talk about, the rubro pathway, which rubro means red, but that is a descending one. So it's rubrospinal, meaning it goes from the red nucleus down to the spinal cord. All right, so visceral sensory pathways are, of course, going to originate in ventral cavity organs, and these are interoreceptors, so they're detecting stimuli and changes in stimuli inside of your body as opposed to stimuli coming from the outside. So I already mentioned nociceptors, which are for pain, and then, of course, thermoreceptors are for temperature. Tactile receptors are obvious. These are two that might not be as familiar to you. Baroreceptors detect stretch of hollow organs and thereby the pressure in the hollow organ. And then chemoreceptors detect chemicals. So, for example, chemoreceptors are going to detect things about your blood pH or salinity or osmolarity, for example. They can also detect things like dissolved gases. So chemoreceptors are for those things, but nothing that you detect consciously. So we can see these are going to travel in autonomic nerves. So we'll talk about the autonomic nervous system in the next PowerPoint. But for now, just know that they travel in autonomic fibers. And that is going to be taken care of by the ventral ramus primarily, because most of your anterior ventral innervation and spinal nerve components are connecting to your abdominopelvic cavity and therefore the viscera. But also, of course, body wall and limbs. So there is a little bit of positional information as well. So that's going to travel in the ventral ramus, and then the interoreceptors travel, oh look, through the rami communicantes. Remember that? And in your model, these were encased in the same epineurial sheath, so you couldn't see the difference, but now you can. So it's going to cross through the rami communicantes and up into the dorsal gray horn of the spinal cord via the dorsal root and ganglion, and it's going to arrive at the lower portion of the dorsal gray horn. So again, since these two things are very close to each other, if I have an visceral organ pain, that might leak over action potential wise or neurotransmitter wise to the neighboring somatic sensory nucleus, resulting in me perceiving pain where it is not. All right. So there's two options for visceral sensory pathways, depending on where the visceral sense is occurring. So abdominopelvic viscera, which are obviously lower, these are going to enter the spinal cord via the dorsal root, so pictured here. So this pathway, but there are viscera that are autonomically controlled in the head and neck. And so those are obviously going to skip the spinal cord due to their position. So they enter the brainstem via the 5th, 7th, 9th, and 10th cranial nerves, which you should know the names of. So I'll give you a little hinty hint here. Vagus, for example, that shouldn't surprise you since you know that it innervates most of the abdominopelvic viscera. And also the thoracic ones, so the thorax, including lungs and heart, and other aspects of the thorax. And then we've got mouth, pharynx, and larynx as well. So Really, it just depends on the position, right? So if you're in the head already, you don't need to do the spinal cord. And then the thoracic viscera is a bit of an exception. And it's a special exception that has to do with sympathetic innervation specifically. So we'll talk about that later. So visceral sensory pathway destination. So where does the visceral sense arrive if it arrives not in the spinal cord in this case? Because we already established that lower visceral senses arrive at the dorsal gray horn of the spinal cord. So where do the upper ones arrive in the brainstem? So they arrive at the solitary nucleus, which is this little guy that's like hidden behind the olivary and the reticular formation. But if you turn it, you can see that it's this one right here. So next to the nucleus cuneatus and nucleus gracilis, which remember are carrying somatosensory information, but visceral sensory information is the solitary nucleus, and there's one per side, so it's not only on one side, there's both, but it's nestled in right there. So because of its proximity to its neighboring respiratory rhythmicity center and cardiovascular center, and the cardiovascular centers contain cardio, acceleratory, and cardio inhibitory nuclei. So those are going to either speed up or slow down your heart rate. So the position of the nucleus, or the solitary nucleus, basically allows it to send impulses to its neighbors to initiate decisions and changes based on the sense being received. So for example, if I detect that my blood pressure has dropped, that impulse, so a loss of blood pressure, so baroreceptors in my arteries are going to detect that my arteries are not as stretched as they should be for my blood. And so they're going to say, hey, our blood volume is low, or at least our blood pressure, we're not sure. But the solution to that is to speed up the heart, so that information would pop over to the cardiovascular centers, specifically to the cardioacceleratory centers. And these ones are not going to go to the cerebral cortex because they don't need to. Your brainstem is really important because it coordinates all of this stuff and sends efferent information on its own to mediate and modulate the function of the organs that are receiving sensory information in this particular pathway. Another example would be, let's do a chemoreceptor example too, so you can kind of consolidate what those receptor types mean and do. So I just gave you an example of baroreceptors. If it was a chemoreceptor change, let's say we had a increase in blood pH. Actually, no, let's do drop in blood pH. So drop in blood pH. That means the blood is becoming more acidic. That is evidence that CO2 is accumulating in the blood for whatever reason. So CO2 plus H2O becomes carbonic acid, becomes a proton, which is the unit of acidity, and then bicarbonate. So if I have a lot of CO2 in my blood, it's going to drive the reaction this way towards the formation of protons, and that's going to drop my blood pH. And that information is sent to our solitary nucleus, and the solitary nucleus is going to adjust our breath rate to scrub CO2 out by breathing it out, and that's going to bring the pH of the blood back up to close to neutral where it should be. So we'll get more information about that on next term because we talk a lot about this equation and breathing, but it's important for you to build up to that by knowing that the solitary nucleus is going to receive those senses and then act accordingly depending on which sense it is and what needs to happen in response. All right, so motor pathways. Visceral motor pathways, again, we're going to do in chapter 16 because those are autonomic nervous system functions. But somatic motor pathways are going to include both planned actions but also reflexes. So we talked about reflexes earlier, and then planned actions are, of course, movements that you intend, right? So depending on whether it's a reflex or a planned action, the pathway looks a little different. So let's go have a look. So motor pathways are, of course, going to be descending, right? So they begin in the primary motor cortex, which is on the precentral gyrus. And of course, they end in skeletal muscles, which we consider to be voluntary functionally. So this is easier than sensory because there's only two. There's in the upper motor neuron, so that's going to be a pyramidal cell in the cerebral cortex of the precentral gyrus. And then the lower motor neuron, which is either a nucleus of the brainstem or the ventral horn of spinal cord. So that's real simple. So why would some be in the nucleus of brainstem? Well, cranial nerves. So for example, motor control of my tongue and pharynx, glossopharyngeal, right? And hypoglossal as well. So that's just one example. There are many others, but those are going to be organized in nuclei of brainstem, etc., because they're not at the spinal cord level yet. And then lower ones, of course, are going to be ventral horn of spinal cord, specifically the ventral gray horn. So 3 motor pathways. One of them is named sensibly, so it tells you information about what it does and where it goes, and the other 2 are just named for their position, which is unfortunate because that doesn't provide you with very much information at all. So corticospinal is going to be cortex, obviously. Precentral gyrus, and then spinal cord, right? And then we have medial and lateral, which we'll also talk about. But their names don't include much information, which is dumb. Okay. The activity of these motor pathways is monitored and adjusted by both the basal nuclei and the cerebellum using proprioreceptive information, but we're not going to talk much about the interconnectivity there, just I'm mentioning for the sake of information that the motor pathways are changed and augmented and influenced by proprioreceptive information and output from those 2 regions. Okay, so we have the anterior corticospinal right here. And then we have the lateral corticospinal here. So let's go investigate what all these mean and what they are. So these ones are considered medial and these ones are considered lateral. So medial and lateral portion, again, doesn't explain much about where it is and what it does, but they're both corticospinal, so you at least get that much, but it doesn't include any information about like their origin point, or I mean, excuse me, not their origin point. It doesn't include specific information about the destination in the medial or lateral name, just that it's corticospinal. So let's go look at what those destinations are. So the corticospinal pathway in general provides you with voluntary control over your skeletal muscles. So you form the motor program in the motor association area, and then you pass the motor program over to the motor homunculus on the precentral gyrus, which contains the pyramidal cells, which are the upper motor neurons that originate and have their cell bodies in the primary motor cortex, and then they send tracts down and out to go off to their appropriate synapse location, either in the brainstem or in the ventral horn of the spinal cord. So these tracts form the pyramids of the medulla. So if you're wondering why those were there and why you had to learn them, that's why. The shape of the tracts on the front, which is obvious on models and on an actual medulla oblongata, is there due to these pyramids on each side descending but also crossing over. So we have the decussation of the pyramids here, which is where a great number of those tracts decussate, but as you can see, not all of them. So if we look closely at the bottom of this image, we can get a little preview and see, oh, the anterior corticospinal tract travels anteriorly before it decussates. So that is going to go straight down, pass through, not decussate in the pyramids, but instead in the anterior great commissure. And then the lateral corticospinal tract decussates in the medulla via the pyramids. So there's a different decussation location depending on whether it's the anterior / medial or lateral corticospinal tract. So we've got the lateral and anterior, which is also medial. And then the corticobulbar tracts, which are their own thing. So let's check those out. So remember I said lateral corticospinal tracts decussate in the pyramids, cross over, and then excite lower motor neurons in the ventral gray horn. The anterior and also medial corticospinal tracts descend, and they do not decussate in the pyramids. They decussate in the anterior gray horn of the spinal cord, and then again synapse at the same location. So they decussate to the other side, and then they synapse on the cell bodies of motor neurons that originate in the spinal cord and go off to effectors. And then if you'll notice, the corticobulbar tract, this is the one that's associated with cranial nerves. So it's going to extend to skeletal muscles and motor nuclei of cranial nerves at both levels. So in the cerebral peduncle and also inferiorly, some of them in the upper medulla, depending on what motor control needs to happen. So these are going to be associated with cranial nerves, the motor ones specifically. And then the other two are going to be associated with lower skeletal muscles away from the head. So anything from, the neck down, basically. So similar to the sensory homunculus that I showed you, there's also a motor homunculus, which is terrifying. So that it looks similar. So we have giant hands, a really big face, and then skinny little arms and legs. So remember, the sensory map was according to how many receptors were in the particular limb. So your hands and face have a lot of receptors. Similarly, the motor cortex map has differential sizes, but the largeness or smallness is not determined by receptors. It's determined by the number of motor units. So remember, let's take a moment to redefine motor unit. That is going to be the fibers, which are, of course, cells, in a skeletal muscle that are all innervated by same lower motor neuron. So the greater the number of motor units, the larger the cortical area is. So we can assume by that the hands and the face have lots and lots and lots of motor units, and then the legs, arms and feet, and the trunk have very few motor units. So that's what this homunculus is demonstrating. And if we think about it, muscle groups with the largest cortical area and therefore the most motor units are going to have the most precise control. And those include fingers, thumbs, so you can manipulate tiny objects, you can play the piano, all kinds of fancy hand motions. And then, of course, we can, in a coordinated fashion, vibrate our vocal cords, change the pitch of the vibration, change the shape of our buccal cavity, and what our tongue is doing in a particular sequence, and that is how we produce speech. So lots of motor units involved in that as well. So just because a muscle is big doesn't mean it has a lot of motor units. In fact, the big muscles don't, because typically you either need to activate most of the muscle or you don't need to activate it at all. And you can recruit more motor units to do things like, continue to hold your arms in the same position as they're holding more weight. So if I was holding one cinder block uh, and then you put another cinder block on top of it, I wouldn't change position, but I would be recruiting more motor units from my uh, elbow flexors, but they still don't have as many as these very delicate and precise motor areas associated with manipulating objects and with communication. So here are the two scary homunculi together, sensory and motor. One thing I do want to point out, just real quick, because it's kind of glossed over, which in your textbook, which is a little bit of censorship actually, which I don't think is appropriate, but it does happen. So you might have noticed that on the sensory homunculus, the genitals are also small in the cortical representation. And this doesn't make any dang sense because, as we know, because our genitals are part of our sexual response organs and they experience lots of sensations, the genitals have lots and lots of receptors. So why aren't they illustrated the appropriate size given the density of receptors that exist there? So specifically, these receptors are going to exist in the glands, penis, and the clitoris, and also just accessory sexual structures to those two things. There's also stretch receptors and pressure receptors in addition to pleasure receptors that release oxytocin via these. But the sensory homunculus doesn't really reflect that, which is odd, but I'll explain. So the primary textbook publishers for anatomy and physiology in the United States are McGraw-Hill and yours, which is Pearson. There's also Elsevier and some other ones, but the big ones are these. And it's in their best interest because they're a publishing house to sell lots of textbooks. But there is a board of people in each state that are responsible for banning books. So banning books means not in libraries, and it means no textbook adoption by state-funded colleges, universities, and also high school and lower school as well. So in more conservative states like Texas, Texas is huge. Texas is both very large and has the potential for lots of textbook sales, and it's also conservative. And so, in an effort to make sure that they can sell books in large places like Texas that are also conservative and not risk the book getting banned and the loss of revenue from the banned books, sexual processes and aspects of human sexual function and sexuality are downplayed in your textbook because the publishers don't want to offend the banned book committees and lose the revenue by getting their books banned. So there's several places in your textbook where there is kind of a anatomical and physiological oversight that, if you think about it, really should be present but isn't, like the sensory homunculus genitals I showed you. And this is the reason for it. So this is a version of censorship, which I think sucks. Boo. If people are trying to learn physiology, that includes the physiology of sexual reproductive organs. And so I'm just making you aware, even though we don't think of textbooks as being subject to censorship, they are. But most of it is done quietly and in back channels, so you don't notice it. So um, hopefully someday we'll get past that and we can just have books that include all of the necessary information to understand something, regardless of whether it makes people uncomfortable or not, because like good clinicians need to know those things. Okay, so we talked about the corticospinal tracts. We've got anterior and lateral. So these are visible as the pyramids. So, oops, let's make this little dot smaller. So this and this, those little sort of baseball bat shapes on the front of the ventral medulla. And the lower motor neurons are in the ventral horn of spinal cord. So these are going to innervate, of course, our torso and limbs. The corticobulbar tracts, by contrast, are going to control the muscles in the head and neck, and therefore they're going to synapse in nuclei in the brain stem, so the pons or the medulla, or even, in some cases, the midbrain. And so these are going to be present in nuclei because nuclei are cell bodies surrounded by white matter. And since we're still in the brainstem, that is the only place where they could synapse. So we know that cranial nerves are associated with nuclei where synapsing occurs and processing occurs. So these nuclei include nuclei that receive input from the corticobulbar tracts. So these are going to be associated with most of the cranial nerves that stimulate muscles in the head and neck and face and mouth. Okay, so the medial and lateral pathways. These control skeletal muscle as well, but instead they're used for reflexes. So I've been mentioning that the anterior is also medial. And the reason I mention that is because we've got the reticulospinal and vestibulospinal tracts traveling medially, but they're also anterior from a positional perspective. So the anterior corticospinal tract is here, and then we also have the reticulospinal and vestibulospinal pathways that also stay medial and anterior, and those are going to originate further down. So reticulospinal and vestibulospinal refer to the vestibular system and the reticular the reticular formation respectively. And there's also the tectospinal, which is up here. So this area of the midbrain is called the tectum. And this is a place where certain visual reflexes and other things are coordinated. So we'll talk about that. So these are going to be controlling muscles in the neck, trunk and proximal limb primarily. And then the tracts are kind of divided by both their location and association, tectum versus vestibule versus reticular formation, and also their functions. So the vestibulospinal tract, unsurprisingly, receives input from the vestibule via cranial nerve 8, which is our vestibulocochlear nerve. And so that's going to, of course, keep track of where your head is in three-dimensional space, and also if it's moving angularly or if it's moving linearly. So remember, we've got the utricle and saccule, which are for linear acceleration, and the semicircular canals, which are for angular acceleration. And so that input is going to inform the action of the vestibulospinal tract. So there's a vestibular nucleus, which is going to control reflex muscles of the neck, head, eye, and proximal part of limbs. And these are going to, for example, for eye, visual changes to maintain balance or orientation, as well as the head and neck and the upper limbs as well. So things like putting your arms out to sort of stabilize yourself in response to the feeling of falling down. An example of eye, head, and neck, specifically eye, is if I put you in an office chair and I spin you around and around and around and around and around, and then I suddenly stop you after a period of prolonged spinning, what you're going to have experience is called nystagmus, which is the rapid movement of your eyes back and forth. And that's because while you were spinning, your vestibulospinal tract was trying to make your eyes pick a spot to orient to that's stationary. So as you turn around, you are looking at a spot, or trying to anyway, using your rectus muscles of your eye. And then when you spun around, you're trying to find that same spot again. So that's a phenomenon that is useful if you're rotating to keep oriented. Ballerinas do it, for example. But nystagmus is the muscular action that too much of that results from. So these are going to basically just adjust all of those muscles to in response to a change in head movement that activates the vestibule. So that's the sort of relationship there. Anything involving imbalance related to head position is going to be coordinated motor-wise by the vestibulospinal tract. So the tectospinal tract I mentioned was in the midbrain. So the tectum is right here. It's underneath the corpora quadrigemina. And this is an interesting one. So it receives input from the corpora quadrigemina, which you might remember coordinate reflex in response to audio or visual stimuli. And so the tectospinal pathway is going to receive input from the corpora quadrigemina about sudden sounds or sudden visual changes, and then run from the tectum to the muscles of the head, neck, and upper limbs, which allows you to reflexively orient towards unexpected visual and auditory stimuli. So jump, turn head, orient toward stimulus. So it starts in the corpora quadrigemina, but the outcome is coordinated by the tectum and the tectospinal pathway. So the reticulospinal pathway, as the name suggests, runs from the reticular formation, which in the central nervous system is very diffuse, so lots of locations. And then the reticular formation basically converges in the brainstem to form this long tract here. So this has lots of different outputs, including some muscles of eye movement for specific things, REM sleep, rapid eye movement sleep, that's coordinated by the reticular formation, but there's a lot of different outcomes, so we're just going to kind of summarize it. So eye movement is one, muscles of respiration. So your respiration is very different depending on whether you're sleeping or whether you're awake. So for example, deep, slow breathing is characteristic of sleep. Shallower, quiet breathing is characteristic of wakefulness. So this kind of accounts for some of the eye and respiratory phenomena that we observe as humans transition from a state of wakefulness to deep sleep. All right, so I mentioned rubro earlier, and it meant red for the red nucleus. Now we're at that point. So the red nucleus is right next to the substantia nigra in the midbrain. The substantia nigra is, of course, black because it means black substance, and the red nucleus is actually red. It looks red. So this runs from the red nucleus, where cell bodies of upper motor neurons live, and in response to input from the cerebellum and cortex, runs from the red nucleus to the cervical spinal cord, and the cervical spinal cord gives rise to the brachial plexus, which innervates your arm. And this provides basically reflex actions to the distal upper limb muscles specifically. So muscle tone and precise movements. So remember, our hands have a lot of motor units, and especially for delicate object manipulation, tone is important because you don't want to like grip a tiny thing too hard. So the micro adjustments in pressure that you make or other things are going to be coordinated reflexively by our red nucleus. So one of the reasons for this is, of course, so distal upper limb is forearm. So we just went over in the muscle video I just made the flexors and extensors of the forearm and how many there are. One of the reasons for their numerousness is because we need a lot of motor units and a lot of different muscles to coordinate all those precise actions. And so the red nucleus helps with that. Okay, so this is an FYI, just, oops, fine-tuning of motor control. So you're going to want to read the section titled Monitoring Role of the Basal Nuclei in Cerebellum. So this is actually an FYI, meaning I'm not testing you on it directly, but basal nuclei get input from the substantia nigra to monitor and take care of smoothing of movements. And so these are all going to play a role in the specific part of the case study that I assigned you. The one concerning dopamine, Parkinson's, and addiction. So that's a big hint, and this is one of the book sections where you're going to find answers to help you complete the case study. So this is just me saying the case study is where this assignment, or this the content rather, is going to be assessed, not in the test. All right, and that is all for chapter 15, Pathways. So I hope you enjoyed it and hope it was interesting and understandable, and I will see you in the next video, which will be about the autonomic nervous system, so parasympathetic and sympathetic. So as always, thank you for your attention, and I will see you in the next video.